Assalamualaikum. My name is Husna Jurrat, and I am doing my PhD from Government College University, Lahore. And I am glad that I have been offered a chance to present my research work on International Brain and Biomedicines Conference, IBBC-2023. Thanks a lot for giving me this opportunity. The topic of my research, MPhil research, is promoting effect of azadi recta indica on mice peripheral nerve regeneration. Problem statement. The problem is peripheral nerve injury affect millions of people. Yes, it is affecting millions of people. Over 200 million people per year are affected by PNI, and 13 to 23 persons per lakh in developed countries are affected by peripheral nerve injuries, and injuries resulting such injuries often result from accident, trauma, and surgical impairments. But unfortunately, only 3% patients are admitted to trauma centers. Introduction. Peripheral nerve injuries affect the sensory and motor functions because, for example, in this diagram, we can see that these are sensory organs and these are motor organs, motor and muscle fibers. These red color nerves are called the motor nerves because they are sending messages, messages, impulses to muscles, and these are known as nerves. These are known as nerves, and they help they are responsible for sensory functioning. And any disruption in these, any injury in these nerves may affect the proper functioning, motor and sensory functioning of cells that affect the quality of life as well. Another problem is nerve regeneration rate is very slow, and thus it affects the sensory and motor functioning of nervous systems. On average, it was reported that any damage or injury in nerves require around 800 days to recover about 800 mm, which means on average per day only 1 mm nerve regenerates. That is indeed a very small amount. So it affects the quality of life, and a research was conducted in order to enhance this intrinsic rate by using some medicines. Previously, surgeries as well as the other treatments were using to cure PNIs, but these FDA approved treatments were quite expensive. So, accelerating axonal growth can speed up the functional recoveries, and traditional medicinal plants are being investigated for promoting effect on recovery process of peripheral nerve injuries. As shown in one study, this is a control group, this is mushroom extract treated group, and this is a medicinal derivative of the medicinal plant. It was given to the organisms with crushed mouse models, and increased reflex responses were observed in treated group. Thus, we can say that these axonal growth can be speed up by using some TCMs and other organisms and other medicines. Medicinal plant and derivatives and nerve regeneration. There are many studies that has been conducted so far to find the nerve regenerative potential of many medicinal plants and their derivatives. So the study used the directa indica leaves extract to find its regenerative potential, nerve regenerative potential. The directa indica is the common name of, it's a scientific name of neem. It is a potential candidate for nerve regeneration due to some primary reasons. The one of them is, and it has antioxidant properties. It has anti-inflammatory properties. It can show anti-apoptotic potential, and immunomodulatory features are also one of the key features of the directa indica. These potentials, these properties of neem leaves has been reported in many studies as shown in this neuroprotective effect of the directa indica, a standardized extract in partial static nerve injury in rats, evidence from anti-inflammatory, antioxidant, and anti-apoptotic studies. So the aim and the objectives of our research were investigation of regenerative potential of AI extract on the peripheral nerve regeneration through the behavioral test, and then investigating the histological basis of enhanced axonal regeneration in sciatic nerves, and then unveiling the underlying mechanisms of regenerative effect of AI. Whole experimental design was this study, for example, this is a diagram showing sciatic nerve. This is the sciatic nerve. The white thread-like material is the sciatic nerve of the mouse. And in this figure, we show that a sciatic nerve crush injury was performed. This nerve was pressed for 30 seconds to give a injury to nerve, and uh, it was then trained by for pinprick assay, toe spread assay, as well as SFI. About 24 mouse were taken from Government College University's Animals House, and they were trained for all these three tests and was pre-treated with or AI methanolic extract. Then on the day zero was these mouse were observed was um, received a crush injury on 0 day, and from day 3 to 29 behavioral test was performed to find the functional recovery. Pinprick assay and toe spread assay were performed on daily basis from day 3 to 29 after peripheral nerve injury, and SFI was performed once in a week from day 3 to 29. And there's baseline line reading observed before giving crush injury was compared with the recovery scores to find the final graphs and to estimate the recovery potential of the directa indica. Then histology was performed, sciatic nerve extraction. This is a picture showing sciatic nerve extraction. This was a white thread-like material which was previously crushed. It was then harvested in this way and then preserved in sciatic nerve is preserved in PFS solution for histological analysis. Histological analysis of gastrocnemius muscle was also performed. Some chunks of gastrocnemius muscles of every mouse was taken, and it was observed in 24 well microteacher plates in having 4% PFA solution for preservation. After that, histology was performed and micrograph was taken for interpreting the results. These were the results of our study as the director indica treated mice weight profile. The weight of all the mice during the experiment was measured, and it was found that there was no significant difference or instant variation via abrupt changes in the weight of every mouse, which shows that the mouse will remain healthy throughout the experiment. Later on, this was the result of pinprick assay. The pinprick assay showed that the mouse receiving methanolic neem extract at 500 mg per kg shows a fastest recovery as compared to the control group, and similarly, the difference between the recovery rate of ME methanolic extract of 5 to 50 mg per kg and vehicle was also observed. Thus, we can say that the directa indica treatment enhanced sensory functional recovery showing sciatic nerve crush injury. This was the result of toe spread assay. The toe spread assay shows that till 7 days from injury to day 7, there was no motor recovery observed in all three members of the groups, but later on they started recovery, and from day 11 to day 7 in 250 and 500 mg per group, fastest in recovery was observed as compared to vehicle group, but this was a mild recovery. Not a significant difference was found between all these groups. The director indica treatment enhances motor functional recovery following sciatic nerve crush injury. SFI scores. SFI score shows that this was a baseline reading, and again the AI treated, यानी कि as a director indica treated mouse shows fastest recovery from week 2 as compared to the vehicle group that started recovery from 3rd week. So it fastens the nerve regeneration, and we can say that the AI treatment, AI indica, yani AI leaves extract treatment can improve the nerve regeneration rate in patients receiving sciatic nerve injury. To further validate the results, histology was performed in the regenerative sciatic nerves, and the result shows that the myelination thickness was greater in 500 mg per kg treated group in as compared to 250 mg per kg group, but as compared to vehicle group, the myelination was denser. The intensity was greater in 250 mg per kg group. This shows that, as shown in this figure, vehicle group has only 20,000 myelin density. AU may is AU are its units, and similarly 250 mg per kg shows 40,000 myelin density, and 500 mg per kg shows 60,000 AU myelin density, which shows that the myelin regeneration, the myelin thickness was greater in AI treated groups. Similarly, enhanced motor axon regeneration and innervation was observed in 500 mg per kg group. It has more fibers reinnervation as shown in this figure, and in the 250 mg there were fewer myelin reinnervating muscles fibers regenerating in 250 mg per kg, and vehicle group has no such vehicle groups, no such reinnervation. So the result of this study was found that azadirachta recta indica enhances restoration of sensory function recovery in mice using unilateral sciatic nerve crush injuries, and azadirachta recta indica treated mice shows an improved but non-significant improvement in mouse treated with AI in motor functional recovery, and azadirachta recta indica administration and 250 and 500 mg/kg do not show dose-dependent effect on peripheral nerve regeneration. And azadirachta indica may be used as an alternative complementary medicine, so further investigation is required. For future direction, in vivo and in vitro investigation of azadirachta indica treatment on expression of regeneration associated genes at mRNA level can be performed, and estimation of azadirachta indica antioxidant potential sciatic nerve injury models can also be performed to further validate results. Thank you.